In this chapter, we shall look at the properties of proteins as enzymes. So, as an overview, we will review kinetic and thermodynamic aspects of reactions from your general chemistry, the mathematical description of enzyme kinetics, also from your general chemistry, and then we shall look at enzyme-substrate interaction, some examples of enzyme-catalyzed reactions, and then the Michaelis-Menten approach for enzyme kinetics, and then we shall look at inhibitors. So we shall explore the nature of enzymes. Enzymes are effective biological catalysts. And catalysis is possibly the most important function of proteins. This is not the only function of proteins, but possibly a very prominent function of proteins. All functions of proteins are necessary for the well-being of the cell and for multicellular organisms, the whole organism itself. Because virtually no reaction in the cell occurs without enzymes. Think of all the chemical reactions, metabolic pathways, or that you can think of, okay, to make your body function. An enzyme is involved in each of those reactions. And there's probably tens of thousands, hundreds of thousands of different reactions. One of them not working will result in an impairment of the ability of that organism to live. Okay? And by nature, almost all enzymes are found to be proteins. There have been some exceptions, okay? Just like everywhere else, there will always be exceptions. So some ribozymes, okay, ribo from the ribonucleic acid, you may have heard of RNA, and the combination of function like enzymes, hence ribozymes. An RNA that function like an enzyme. If we talk about non-enzymatic catalysts, inorganic catalysts, they enhance the rates of reactions by a factor of 10 to the 2 or 10 to the 4. That's 100 times fold or 10,000 times fold as much. No? So you may think that's fast. Yes, they are fast. However, they pale in comparison to the effectivity of biological catalysts. These enzymes enhance rates of reactions by a factor of up to 10 to the 20. Can you imagine that number? Okay, so analogy, say non-enzymatic reaction, you can walk from your living room to your kitchen in, I don't know, 5 seconds, 10 seconds. If you're now you have a non-enzymatic catalyst in 10 seconds, you probably reach, say, from ADU to GMOL, maybe. And then for enzymatic reactions, you probably go in 10 seconds from ADU to the moon or something like that. Okay, so just imagine how fast and efficient biological catalysts are. And not only that, enzymes also exhibit specificity. Okay? So it may act on one enantiomer, but not the other. It may act only on alcohols, but not aldehydes. It may act on amides, but not esters, or things like that. And another thing that makes life possible is that these enzymes can be fine-tuned. What do you mean by this? That means they can either be speeded up or slowed down depending on the metabolic needs of the cell. Amazing, right? So here's a comparison of catalysts. Okay, so let's look at the reaction of hydrogen peroxide, okay, agua oxygenada, if you will. You may find this in your household. Okay, hydrogen peroxide undergoes decomposition into water and oxygen. Okay, so I highly suggest that you go to your medicine cabinet and then look at the decomposition of hydrogen peroxide into water, okay, and oxygen. Okay, so see the oxygen, the levels of oxygen rising. Maybe there will be bubbles. Okay. Very slow, right? Now, if you can find a catalyst, I don't know, something lying around, you can try various inorganic materials and see whether a pinch of that would make the hydrogen peroxide decompose much faster. Okay, in the lab we can take, say, manganese dioxide. In this table, it's a platinum surface, but again, if you have platinum lying around, you can try the reaction. Okay, and you will see that the hydrogen peroxide will bubble much faster compared to just a simple hydrogen peroxide that you may see at home. Now, another one is catalase. Okay, so if you can get hold of catalase, expose that to hydrogen peroxide, and you will see it bubbling much, much more vigorously compared to, say, if you have platinum or manganese dioxide, as the case may be. Okay, have you done that? Okay. Actually, you've done this last experiment, probably done this last experiment yourself. If you've ever wounded yourself, cut yourself, and you were told to disinfect the wound by adding hydrogen peroxide, notice how vigorously it bubbled. No? So that's due to the presence of catalase in our bloodstream. Okay, because catalase, its main purpose is to actually, decompose hydrogen peroxide whenever it forms in our cells, and hydrogen peroxide does form in our cells. Amazing, right? Now, what would affect the effectivity of enzymes? Well, enzymes are simply proteins. So, just like as was pointed out earlier, enzymes are also affected by changes in temperature. Okay, so reminder what happens with temperature. As you increase the temperature, the kinetic energy of your molecules increase. However, since proteins are peptide chains, okay, all this string bundled up together increases in kinetic energy. At some point, some of the intermolecular forces that hold them together can be overcome, and you denature your protein. Another is pH. Again, reminder, it was already mentioned before that, again, some of the side chains in your polypeptide chain could be carboxylates or ammonium, which means that it can either be protonated or deprotonated depending on the pH, and it will disrupt the intermolecular forces that holds them together. So it's the same for catalysts, for enzymes. Okay. So here's a plot of the percent of maximum activity of a particular protein as a function of temperature. If you recall from your general chemistry, okay, normally as you increase the temperature, the rate of reactions also increase. So it is the same for proteins. However, a peculiarity again of proteins, enzymes, is that at some point you now start to break down some of the intermolecular forces that hold them together, changing the shape of the protein and then changing also its effectivity. Okay, so reminder. So this is another illustration or hint about the structure-function relationship present in biomolecules. The curve for pH is probably similar. No? So there's an optimum pH at which enzymes work, and physiological pH is usually or is around pH 7. Okay, so when you hear physiological pH, it means pH 7. Most enzymes, I probably expect, would be at their optimum activity at pH 7. However, there are enzymes, especially you can probably think of our digestive enzymes in our stomach, where the conditions are acidic. So the enzymes, the digestive enzymes present there, will work best at acidic pH. And then at other conditions, okay, so I think in the intestines, we have, it was mentioned that the conditions there are more basic. So the enzymes there have probably optimum pH at basic pH. Okay. So before we move on, let's discuss nomenclature of enzymes. This is not mentioned at all in Campbell. Okay. So enzymes, okay, have a common name. Okay, you probably know, familiar with some of them. It's usually named after the substrate or the reaction as the root word plus ase as a suffix. So for example, sucrase. Sucrase, can you guess the substrate here? Yeah, sucrose. Okay, so it's this enzyme that acts on sucrose. Or the type of reaction. We will encounter this later. DNA polymerase. Okay, so the key word there is DNA as a polymer. Okay, so this enzyme polymerizes our DNA. Some of them have historical names, like from biology, the digestive enzymes, okay, trypsin and pepsin. Again, as the biologists among you may probably be more familiar with these ones. However, okay, with a multitude of enzymes, uh, you really cannot have just a reaction. Okay, for example, there are many types of reactions, and they may also have enzymes, say, that work on sucrose, but catalyzes a different kind of reaction. So you can technically consider it sucrase. However, it may be a different reaction. So you can just imagine the confusion that may arise. So the systematic way of naming enzymes is by the International Union of Biochemistry and Molecular Biology, wherein the reactions that enzymes catalyze have been categorized into 6. Okay, and then the full name is EC a number dot number dot number dot number, which represents the class of enzymatic reactions that it catalyzes, and then a subclass of that type of reaction, and a subclass of that type of reaction, and then finally another number. As I understand it, the final number, I think, is just again by could be a little bit arbitrary, or it could be by the order by which they studied it. Okay, so for more on this, visit the website www.chem.qmul.ac.uk/iubmb/enzyme. So what are the classification of these enzymes? The first three are shown in this slide. First one is oxidoreductases. So a little bit obvious, they catalyze redox reactions. And it's a little bit obvious because they're usually named like lactate dehydrogenase. Okay, so the dehydrogenase is a clue that that enzyme catalyzes redox reactions. Specifically for lactate dehydrogenase, its name is EC.1.1.1.27. The next class would be the transferases. Okay. From the name, they transfer a chemical moiety or a group. No? So it's a part of the molecule. Uh, an example of this would be kinases, also alanine transaminase. Okay. So FYI, kinase transfers a phosphate group to or from usually ATP. You've heard of ATP before. Specific example shown over here, alanine transaminase is EC2.6.1.2. Okay, so can you guess the reaction here? Okay, so it's alanine transaminase, the common name. So it transfers an amino group from one molecule to another. Usually it will be from alanine to something else. Third class, dehydrolases. So from the term, it catalyzes hydrolysis reactions. For example, chymotrypsin. We've been encountering chymotrypsin many times already. And another one, pyrophosphatase. Okay, the IUPAC name is EC 3.6.1.1. Oh, sorry, not IUPAC, IUMB name. The next three would be the lyases, isomerases, and the ligases. With lyase, it means the breakage of carbon-carbon double bonds. So we will see aldolases later. okay, in the glycolytic pathway. A specific example is pyruvate decarboxylase with a official name EC 4.1.1.1. Isomerases. This transfers a functional group within a molecule. No? So just think of isomerization. Uh, so enzymes that catalyze isomerization reactions are isomerases. An example is aconitase, which we'll see in the citric acid cycle later. And a specific example with a proper name is alanine racemase, EC 5.1.1.1. Okay, so you should be able to guess the type of reaction of the alanine racemase. If you recall your organic chemistry, racemization is the conversion of an optically active compound into something that's non-optically active. No? So reversal of the stereochemistry. The last one would be the ligases. So ligase, or from the word ligate, which means to join together. So it combines two groups together. So example would be the synthetases. We will meet synthetases later. One specific example is glutamine synthetase with the name EC 6.3.1.2. Okay, so now we will review kinetics and thermodynamics from your general chemistry. For the generic reaction, say AA plus BB forming products. Again, small letters refer to the coefficient, and capital letters would be for the generic name of molecules. The rate of the reaction will be given by rate equals the rate constant times the concentration of A raised to some power x times the concentration of B raised to some power y. And then the rate constant is given this equation. A, some activity factor, times E, the natural number, raised to the power negative EA over RT. Recall also equilibrium. Okay, so the generic KEQ equation will be, okay, for a reaction of A plus BB in equilibrium with CC plus DD. Okay, you have this. I'll not read it anymore. You can just view the slide. And then the uh, energy profile of a reaction. Okay, so on the x-axis is the progress of the reaction or time. Okay, so you're going from reactants into products. And then the free energy or the energy involved in the reaction as you go from reactants to the products. Okay, so recall reactants need to undergo some transition into the transition state, and it requires an energy called deactivation energy, after which once it reaches the transition state, it can either go back to being reactants or proceed into the products. You can also visualize the reverse reaction. Okay, the products can be converted back into the transition state and then go back to being reactants. However, notice that again, the activation energy for the reverse reaction is now, in this particular example, much higher than the energy needed for the forward reaction. In contrast, if you now have catalyzed reactions, okay, it involves an alternative mechanism wherein the activation energy is much lower. Since the activation energy is lower, this reaction can now proceed faster compared to the uncatalyzed reaction. And if you notice, the delta G of the reaction is not affected by the presence of a catalyst or enzymes as the focus of our chapter right now. Okay, so this is just for, again, maybe for the interest of chemists. Okay, so it's just a little bit of a background on thermodynamics. Okay, for a reaction taking place at a constant temperature and pressure. Okay, for a reaction, again, A in equilibrium with B, The change in free energy is given by, again, this Gibbs equation, delta G naught equals delta H minus T delta S. Okay, and then the relationship between delta G and the KEQ is given by this equation, delta G naught equals minus RT ln KEQ. Okay, so let's now look at the mathematical treatment of enzyme kinetics. So we have the generic reaction A plus B is converted into products. Okay, so for simplicity, we'll just look at reactions with only one-to-one correspondence, meaning the coefficients for the reactions is equal to 1. Okay, so the rate given by, okay, the negative of the change of concentration of A, because this is our reactant and we expect the amount to diminish. Therefore, to keep the rate a positive value, a negative sign is placed. Okay, and then you have the time. But it should also equal to the rate of the disappearance of B, or the change in concentration of B over time. And then if you are monitoring the product, we expect the product concentration to increase over time. Therefore, there's no negative sign anymore. So the change in concentration of P over the change in time. Okay, so once again, the generic equation for the rate law, the rate will be proportional to the concentration of A raised to some power F this time times concentration of B raised to some power G this time. Okay, so those are just random coefficients. And then if you want to make them into an equality, recall your high school algebra, you remove the proportionality relationship, make it equal, then you have to introduce some constant. Okay, so since we're talking about a rate reaction, this constant is called the rate constant. Okay, so again, arbitrary powers shown over there, F and G. Those are also known as the orders of the reaction, and they have to be determined experimentally. They do not necessarily have a correspondence to the coefficients of the reactants in the balanced chemical equation. Overall order of a reaction is just a total of those exponents. Okay, now for biology, biochemistry, the rate of the reaction is also known as the velocity of the reaction, also known as the speed of the reaction. So, okay, for our purposes, keep in mind the rate, which is the one that you're probably used to in your general chemistry. The symbol is usually R, but for biochemistry, the symbol is for velocity, so it's V. So let's recall the significance of first order reactions. It means that the rate of the reaction is directly proportional to the concentration of the reactant. In other words, if you double the concentration of your reactant, you double the rate of the reaction. For second order reactions, the rate of the reaction is proportional to the square of the concentration of the reactant. In other words, if you double the concentration of your reactant, then the rate is quadrupled, or 2 to the power of 2, okay, or 2 squared. And then for zero order reactions, okay, recall that any number raised to the power of 0 equals 1. Therefore, the rate of the reaction is independent of the concentration of the reactant. So even if you double the concentration of your reactant, the rate of the reaction remains the same. And then we will focus only on first order, second order, and zero order reactions, because typically reactions follow first order, second order, or zero order kinetics.